Now, I'll turn it over to Debra. She's going to give us an update on the hollow fiber system.
Great. So I'm back. Good afternoon. It's my pleasure to talk to you a little bit, as Klaus said, and update you on the additional work that we've done in the hollow fiber system. So we've talked a lot about how CPTR has invested in evidence-based evaluation of drug development tools and methods. And what do we mean by that? The idea is we want to help you as developers, as product development partners, make better decisions and feel confident in the models that you're using as part of your development process. And so, you know, we've invested a lot of time and money in some of the early preclinical models and evaluating them. You've heard me, Dr. Gumbo, even Eric Nuremberger talk over the past couple of years, about the in vitro hollow fiber system itself. And Klaus mentioned we moved this a couple of years ago all the way to regulatory endorsement. We've invested a lot of time and energy with our partner at Baylor Labs since that time to even further improve our understanding about this system, how it can be applied in the drug development process. And we've asked a lot of questions about potential for variability within the model and its overall reproducibility. And so I want to spend time talking to you about those new data today, but before I do that, I do want to give, for those of you in the room who have never heard us talk about this model, a little bit of background to put into context why we've continued to invest so much time and effort into this drug development tool. So just an overview of the talk that I'm going to give today, more so that you can orient yourself with the deck if you decide to take it home after the meeting. So when we talk about drug development tools and methodologies, we're looking across the paradigm for TB drug development, specifically around models and methods that will help us better pull together these new combinations, these new regimens, and test them early in the development process. As you know, in the preclinical space, there are a lot of models out there that have been used and continue to be used to make drug development decisions in this space. Many in vitro models, even more in vivo methodologies. Our question as a CPTR initiative and with many of our partners like NIAID is really what are the best models to inform these critical decisions. For us, we wanted to do sort of a systematic review of these models. We started with the in vitro hollow fiber system simply because of the amount of data that was available for this in vitro pharmacokinetic dynamic method. And we do have a plan to then go look at some of the next stage in vivo methodologies. Eric will talk to you later about a similar type of evidence-based review that we're doing with a sterilizing mouse. But the idea was, how do we ask really important questions about the utility of hollow fiber specifically and feel confident to the place where we'd apply this to very complex regimen development paradigms. So, as some of you have heard, back in 2014, 2015, our preclinical clinical sciences team working group within CPTR took on this evidence-based evaluation of the hollow fiber system. Our plan was to work towards regulatory endorsement if the supporting data warranted that. We were going to use and did use the methodology guidance pathway, both from EMA and FDA, to evaluate this system. Gather all the relevant data. So Dr. Gumbo's laboratory, several others contributed. We also went into the published literature, and we looked at the potential for predictive accuracy of this in vitro tool for clinical outcome. So again, I'll say we chose this model initially because there was much more quantitative evidence and data available for this system, even compared to the mouse model systems that have been applied for the last 4 decades. And then we move forward with a very in-depth assessment of predictive accuracy. I'm not going to go through the details of that assessment because we've covered it in previous workshops and various other venues, but simply to say that just like or similar to this meta-analysis that Radha just talked about, the goal was bringing all of these different data sources on this particular in vitro preclinical model, pulling them together in a data platform, which was provided by CPTR, aggregating all of those data to the standard and then comparing them to the clinical data. And all of that is really important and fundamental to the work that we continue to do with other preclinical models. So as Klaus alluded, and we've talked about before in this forum, we followed that qualification or methodologies approval pathway for both FDA and EMA. And this was back in January, of 2015, that we did achieve a qualification decision by the European Medicines Agency, specifically talking about the application of this tool in the drug development process. And you will see very similar language in that document in the FDA updated draft guidance for TB drug development. And this is essentially what it says. So our team had defined a specific set of applications for this tool in the drug development process. And that's what we call context of use, right? So went through the process, had the supporting data, and the regulatory authorities agreed that we had sufficient evidence to say that the hollow fiber system is qualified for use in drug development programs as additional and complementary tool. So we say this over and over again. We're not looking for any one tool to make all great decisions in drug development. It will be a suite of tools, and this is one that can be used with confidence. It can be used in regulatory submissions, especially for informed design and interpretation of clinical studies. And it's recommended to be useful as follows: to provide preliminary proof of concept for developing a specific drug or drug combinations to treat tuberculosis, to select the pharmacodynamic target, to provide data to support PK/PD analysis leading to initial dose selection for non-clinical and clinical studies, and to assist in confirming dose regimens for later clinical trials, taking into account human PK data and exposure-response relationships. So we didn't stop at that qualification. We knew there was a lot of work left to be done in really understanding the potential for variability in what is, as I'm showing you here, a pretty complicated and highly engineered system, right? These are not straightforward plate-based assays. So we as a team wanted to understand the sources for variability in the system and also start asking some really good questions about reproducibility of the model if it were to be applied by different teams and or different laboratories. So again, for those of you who aren't familiar with this tool, which we've talked about so much, this is an in vitro pharmacokinetic dynamic system. It's essentially a very sophisticated chemostat system without the liability of diluting bacteria and other problems that you have in that one compartment chemostat model. It's actually a two-compartment model of infection, which is shown in the upper right-hand corner. That's the fiber bundles that are actually held in that hollow fiber system. The drug or PK concentrations in the system are driven through a mechanized pump system. You can simulate PK of just about any drug you want, and you can manage multiple different half-lives if you're testing combination drug regimens in the in vitro system itself, which is really important and a nice feature of the system. From a pharmacodynamic perspective, part of the work that we've done in the reproducibility studies that I'll talk about is to look at all the metabolic states, the three major really important metabolic states in the hollow fiber system, to further understand how reproducible we could look at these various states of the organism. I'll say in terms of the pharmacodynamic piece of this model, what's really important is that we can do lots of serial sampling. And so you've heard about throughout the day already the need to have more quantitative data. That is a very hard thing to do in a mouse system. It's very hard to do sometimes even in clinical trials, but you can use this in vitro model for those purposes. So what are the inputs and the predicted outputs of this model? So we can, in this system, input human PK parameters, which is, again, an advantage, as you can understand, over a mouse model system. We know going into these studies, static in vitro MICs or sometimes our in vivo preclinical PK/PD information, the experimental conditions that we can assess, either monotherapy with drugs or combination therapy, as I just mentioned. We can look at these three metabolic states: log growth, semi-dormant, and intracellular, and we know from data already produced in this model and data you'll see here, it's really important sometimes to be able to look at all of these three growth phases, if you will, in order to really understand maximized efficacy of drugs or drug regimens. Outputs: target clinical dose, the rate of killing, the potential for rate of resistance emergence, how can we rate order regimens, onward and upwards. Some of that was mentioned in our qualification document. Some of it is just continued information that we're learning in the application of this tool itself. So now I'm going to move into the new stuff, the really, really good stuff. And to just say this was a tremendous amount of work that was conducted by the Baylor laboratories. So I want to talk to you about the design of these studies and then just give you a flavor of the data, because we'd be here all day if I went through every cross-sectioning of what came out of these studies. So again, to state our objectives, the tool was already qualified and or endorsed in one way or another by regulatory authorities, but that we felt there was a lot more work to be done to characterize the signal-to-noise potential in the hollow fiber system. So again, potential for variability under the different growth conditions. And these were a massive amount of studies that were produced, you know, through January of 2014 through February of 2016. And the design of the experiment, again, very massive in scale, ended up to be 162 individual hollow fiber system units. And so that big engineered system that I just showed you, Tawanda in his laboratory did that 162 times. There were 6 treatment arms within the study: the positive control, which was HRZE, Remox 1, Remox 2, high-dose MRZ, high-dose 3-day MRZ, and the untreated control. All 3 growth conditions that I mentioned to you: log, semi-dormant, and intracellular. And here's the part that I think was very, very important to this study, is that Tawanda's laboratory is scaled enough that he was able to pull together 3 individual teams that studied each of these experiments and each of these experimental conditions, such that it's like reproducing 3 different laboratories. So each of these 3 teams had their own individual principal investigator or leader, plus 4 laboratorians that designed the study. So this helped us to get the first snapshot of what could variability look like between different groups of human beings doing this study. So whether it's different labs or different teams, that gives us a good sense of where we're going with these experiments. Then all of those experiments were performed in triplicate. The experimental questions being asked and the ones that I'm going to show today. So again, I mentioned a tremendous amount of data, as you can imagine, came out of these hollow fiber units. We cross-sectioned those data in all sorts of different ways. But today I'm just going to give you a flavor for our confidence in being able to measure PK of different drugs accurately, that we're able to achieve our targets pretty reproducibly, and that to look at intra and inter-team variability, and then to look at variability and kill rate across those 3 different growth phases. So again, I am just going to barely touch on the data that you're about to see in some of these next slides, but Dr. Gumbo is here, I'm here. We're very happy to talk about these data in more detail at some of the breaks. And so here we're showing you the results of the in vitro hollow fiber system Remox reproducibility studies, showing you that we can reliably achieve our target Cmax and AUC for all of the drugs that will begin on this slide and then will be listed again on the next slide. So isoniazid, high-dose RIF, PZA, high-dose PZA. You can see in that center sort of yellowed out bubble the target that we were seeking to reach, and that we came very, very close in what was observed and measured. And that we can do this with high confidence when we look at these doses being measured across all of those different units, the studies done in triplicate, and also tested across multiple different teams. In this slide, I'm just going on to show you the ethambutol data, the MOXIE data, and the high-dose MOXIE data. Again, in that yellow bubble, you'll see that we achieved the targets we sought to in terms of PK. And so for all of these experiments, as you know, Tawanda and his laboratory will do lots of serial sampling and PK measurement throughout the course of the experiment to always ensure that we're measuring, or we have the amount of drug in that system that we're seeking to experimentally test. So in the next few slides, I'm just going to show you the effect of reproducibility across 3 different teams in terms of killing or slope across time courses. So this first slide is going to be our log phase time course. I apologize, there's one piece of the legend missing here. So that is how we cluster teams, and they're clustered by colors. So again, there are 3 different teams here that are assessing, and you can see the greens line up, the reds line up, etc. So all teams found equivalent results for the different groups tested. The HRZ and high MRZ group showed a marked difference against the untreated control. However, the HRZ and high MRZ groups achieve similar treatment effects, right, compared to one another. The key here on this slide and the message I'm trying to get across is that across multiple teams, across multiple different testers, across multiple different experiments, we have very little variability. And in fact, the most variability we see in terms of kill and slope is in the log phase time courses, which we would expect, right? These are the happiest, most rapidly growing bacteria of the conditions that we test. And so in order to account for this, one of our learnings is that we will do replicates of 4 for all of the untreated controls and likely in the log phase growth, right, to be even more reproducible. So this is the semi-dormant time course. Control, HRZE, and high-dose MRZ. As in the log phase condition, the experimental teams found equivalent results for the different groups. HRZE and high MRZ groups showed a marked difference against the untreated control. However, the HRZ and high MRZ groups achieved similar treatment effects. And given the reduced bacterial activity of the semi-dormant condition as compared to the log phase growth, the overall variability in results was reduced across the groups. So the last condition we'll look at is the intracellular time course. And again, the results were consistent across all the teams. HRZ and high MRZ groups showed a marked difference against untreated control. However, unlike the other 2 metabolic conditions, you start to see the difference in the different treatment regimens. The intracellular experiment showed a marked difference between all 3 groups, favoring the high MRZ regimen. So with these data, again, which are much deeper than what I'm able to present in a 15-20 minute presentation here, along with the work that went into the qualification of the tool itself, we believe we have good confidence on when to apply the hollow fiber system model to provide preliminary proof of concept for developing a specific drug or combination to treat TB, again, to select the pharmacodynamic target, to provide data to support PK/PD analysis leading to initial dose selection for non-clinical and clinical studies, and to assist in confirming dose regimen for clinical trials taking into account the accumulated human PK in healthy volunteers, patients available on exposure-response relationships. So these are some of the places we think we can move to as we continue to generate more data in the system, which is exactly what we're doing now. So we're really moving on to our 3rd stage of work in the hollow fiber system with the Baylor Laboratories, looking at a lot of prospective combinations, which include trying to understand better some of the oxy class of compounds and how they may improve different combination regimens. We're looking at things like PAMZ. We're also partnering with some of the pharma partners that are here in this room who would like to test their compounds in the hollow fiber system model now that it's been qualified, and working towards a path to expand capacity with a 2nd laboratory. We talked earlier today, or heard earlier today, about very exciting compounds coming through the late discovery and into the early preclinical space. And given what we've collectively said is the utility of this model, we would like to partner with the accelerator to help get some of these compounds tested in the hollow fiber system. And so we're working towards doing that. And so with that, I will say again, hopefully I've said it enough during this talk, a special thank you to the Baylor Laboratory. He's probably got 15, 16, 17 laboratorians working on some of these studies right now while he's here attending the meeting with us today. So Tawanda and I would be happy to take any questions either now or during the panel session.